Hello and welcome back. So this is the recap of this week's Open Science, Extremely Open Science stream. And I am going to, as usual, start with a recap of the recap. So to recap, we are trying to understand how small mutations impact estimates of quality, and then potentially to use the fact that small mutations are going to introduce apparent pseudogenes, or which means that things that are pseudogenes in the sequence, but were not really pseudogenes in the original bacterium. So we've been struggling a little bit with terminology. These are we call them pseudogenes, because a pseudogene is a real thing that exists in a real organism that kind of looks like a gene, but it's not. And what we've seen is that in sequencing projects, small mutations in those show up as an overabundance of these apparent pseudogenes. So last week we had started this project, or we had continued actually, and we had written code to generate mutated sequences, and then ran prodigal, and we had seen that what will happen is something like this, where, so for example here, this is the average gene size as the number of mutations increases, and it would go from 0 to 50,000 mutations. That was our simulation, and it would start decreasing, and so just the average gene size, this was starting with E. coli K12, and then we had shown that if you can zoom just in here at the very beginning, so even a relatively small number of mutations, and the average gene size goes down quite quickly, while the number of genes goes up also quite quickly, so even at the very start. But we wanted to see what would happen with checkM2, and so this is what we did this week. That was our main focus this week. And then, as these things take a long time, we didn't really finish on the stream, but I let it run. Finished yesterday, and now today I'm recording the video. This is also why now there's a bit of a lag between the time when I do the actual stream and the recap, and this is for me to just wait for everything to finish running so I can also show you the results. So what we did, and I'm a little bit jumping to the end here, but so this was the main thing that we wanted to work on, was running checkM2, and so we wrote this function that takes a list of files. So actually checkM2 can work with multiple files at the same time, and so we built this directory, this temporary directory, where we put all of the files, or actually links to them, and then we simply call checkM2 with the right commands. So in here, so we cannot install checkM2 on the same conda environment, so we installed it on a separate conda environment, and then we can use this conda run subcommand, where we say conda run in the environment checkM2, run checkM2 predict, right? And then this worked quite well, and this worked a little bit. I'm going to show you the results. I've been preparing this a little bit, so this ran, and as you can see, and then output this quality report. And yeah, so here you can see, I'm just going to show you, you can see that, yeah, so this is what the files, this quality reports looks like. So here's the E. coli K12 mutated 0000, so this is the number of mutations, and then the completeness 100. And so I'm just going to make this a little bit easier to read now. So I'm going to, and this is a little bit of a hairy command here, but I'm going to, so Q with columns adds a column where I take the name, so this is this thing here, and I'm going to just do a bit of splitting on the dot, so to get just the file name without the extension, then splitting on the underscore, getting this last element, and then converting that to an int, and that's the number of mutations. And so now Q has an extra column with the number of mutations, and let's so it's sorted in some weird way, so let's just sort by the number of mutations, so that now Q starts at 0, so this is E. coli K12, and then at 50, 100, etc. And you can see here that when it comes at 100 it's still, here it's 100% complete, almost no contamination. I mean, this should have zeros. This would be 100 and 0. This is, and it works quite well. And then with a few hundred mutations, still 0. But then with a lot of them, the completeness has completely cratered down to 50. So let's look at the plot. Okay, and see if you can see that this is I had shown this before, or I had run this before, so everything is X set X label. And here, so I for reasons that will become apparent in a minute, I've actually normalized the completeness from, so this is normally expressed from 0 to 100, but here I've normalized it to be 0 to 1. So first of all, actually quite nice, and I'm going to email this to Alex, who's the first author on the checkM2 paper. You can see how checkM2 is picking up that the genome is not, becomes increasingly bad. So you can see that it picks up, and by 10,000 mutations, it's already pretty bad. So if we set this, so here normally 95%, so that's about here, is the threshold for a high quality genome. You can see that it doesn't take that much. So the reason oops, sorry, I forgot. So the reason why I normalize this is because now I'm going to put the alright. So I'm going to plot the number, the size of the average gene, also normalized so that it starts at 1 and then it goes down. And so what's interesting here, is that if we now, I'm going to just zoom in at the top. So this is the same zooming in that we had before. So let's zoom in only to 2,000 mutations. So the average size of the gene starts to go down very, very quickly. So even a few hundred mutations you also see, and checkM2 is a little bit slower on the uptake. So eventually it does catch up. So if we have it at 10,000, so you can see it's starting to catch up, and it quickly actually goes below the 95% mark. So if I set, let me just put here, sorry, I'm just there's a slightly nicer way of doing this, but I'm just going to do this way. Yeah, so if I put here a dashed line, so you can see that it eventually crosses, and there. So at this point, it would no longer be considered a high-quality genome, high-quality MAG, if we had a MAG. Right. So this is nice to see. I mean, this plot, the blue plot, is the original plot that we saw in some ways in our real data we were seeing this effect that as we had more indels, we were seeing that checkM2 was reporting lower completeness scores, even though the size of the genomes really hasn't changed. So the size of the genomes in this case, we are removing a few base pairs here and there, but adding some others ones here as well, and in expectation, this the size doesn't change. We're as likely to add a base as to remove a base. So this is nice that we see the completeness drop, even though we don't have less sequence, we just have worse sequence. So and that gets reflected in the completeness. Well, we can also add just for we can add contamination, and so the contamination, you can see it also starts increasing a little bit more slowly, but we also eventually it also shows up as contaminations, which is actually also kind of interesting to see this effect. Right, so this was the main conclusions. I'll be sharing this on the mailing list as well, but we also did a couple of other things. So we checked so one thing we did, and this was very code based, is we just improved our mutation code. So this mutation code, we kept the same thing as before, where we're mutating by calling a function called mutate1, but now mutate1, instead of taking a string and returning a string, takes a list of strings, so the individual bases, and then operates on that list. And this is I was going to say it's much faster, but principally it scales much better, which means that we are going to pay a cost to convert from a string to a list of characters. We are going to pay a cost at the end to convert from that list of characters back to a string. But then each individual mutation is very, very fast, because this means that when we go from 10 to 100 to 1,000 mutations, it really doesn't increase the time that much. And so we kept almost everything stays the same. Obviously, there are some, the results are going to change ever so slightly. So we reran this. And the other thing I've changed here also is I've renamed the output files so that the number of mutations always has 6 digits, starting with leading zeros. I mean, this just makes things a little bit nicer to read, because now when you sort them, 000 comes before 0001, which comes before 0002, and then eventually 0010, as opposed to the alphabetical sorting, which messes everything up. So we did that, and then we created a random DNA just of the same size as, and here we used a couple of different models. So the easiest model is this one, just pick ATCG, completely random. That works. A slightly nicer model, and this is very standard, is to use a Markov chain, where you have to somehow have to choose how you start, but maybe you can start randomly, and then for example, let's say you have size 2, and then you take, so if you start randomly, say 80, now after 80, what is the next base pair? It's not completely uniformly random. There's a bias here for certain patterns, and we learned that from the original input data, and then we just sequence. We just sample from that, and then we did that. So this initial filling of the sequence completely at random, uniformly, and then randomly but informed by the distribution. This is very standard. Actually, here, if you saw the stream, GitHub Copilot helped a lot, but it was one of the cases where it got the right gist of it, but we couldn't just accept its code directly. So we ran this, and I forgot. Random. So the average, it's actually nice, because you can see that as the model becomes a little bit more realistic, so the uniform, it's not. So the uniform, it's 184 is the average gene size, and use slightly more complicated models, the gene size is actually a little bit shorter. But just comparing this to even with 50 base pairs, the average gene size, there's still some signal there. So this was, there's not much to learn here, just a little bit interesting to start to get these intuitions, start to fiddle with it and see. So as I said, and I should have actually saved that. I'll run it again afterwards. I'll save the figures. So this figure where we see that checkM2 really is picking up this this decrease in quality brought around by the indels and by small mutations. That was really nice to see. That was somehow that's where this project started. We still see that for a very small, so there's a couple of things here. So for a very small number of mutations, I know although maybe hundreds of mutations, not that small, but we saw that there was already an effect that genes were messed up, but checkM2 was still not picking that up, and maybe we'll try to look a bit more into that. Is it because the genes are you have messed up genes, but it's still okay? So if you have a gene that's 200 amino acids, and now we've somehow cut down the last 50 of them, maybe the first 150 are still good enough for checkM2 to say, oh yeah, this is the blah 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 hydrogenase. Or we'll try to look a bit more into those kind of questions. Maybe we'll look a couple of other genomes as well. Maybe we'll take a couple of mags and put them through the same pipeline next week. So we'll look into all of that. So in the meanwhile, so subscribe to the channel, or comment below if you have anything. I'll be posting this on the mailing list. I'll be and I'll be seeing you all again, same time, same place next week. Thank you.